Types of speciation. Speciation is the evolutionary process by which new biological species arise. There are two basic ways in which this can happen. A single species may change over time into a new form that is different enough to be considered a new species, a process known as anagenesis. More commonly, a species may split into two groups that no longer share the same gene pool. This process is known as cladogenesis. There are several ways in which anagenesis and cladogenesis may take place. In all cases, reproductive isolation occurs. Allopatric speciation, the most common form of speciation, occurs when populations of a species become geographically isolated. For example, the uplift of a new mountain range might divide an ancestral species into two isolated groups. Once the species are separated into these groups, gene flow between them cease, and each group may accumulate genetic changes that serve to differentiate it from the other, resulting either from natural selection or from random events. If the environment on either side of the barrier is different, natural selection may favor genes that produce different traits on either side of the barrier. Even if the environment on either side of the barrier is similar, it may happen that the separated populations might experience different mutations that may persist differently in their respective environments. Also, by chance, some versions of genes may become common in a small population, while others would disappear through an evolutionary process called genetic drift. If the two groups never come into contact again, they will almost certainly accumulate enough differences over time to become separate species. However, if they expand their ranges and come back into contact, some other mechanism must act to preserve the differences they evolved during isolation. Traditionally, this mechanism is known as reproductive isolation, which can impede the gene flow between the two groups, thereby allowing the accumulated differences to persist. Reproductive isolation can operate prezygotically, pre-mating, or postzygotically, post-mating. Prezygotic reproductive isolation prevents fertilization from taking place. It may either be that members of the two groups breed at different times of the day, or different times of the year, or in different habitats, or that the separated members may have developed mechanical differences that prevent copulation. Moreover, even when copulation does take place, the two groups may have become chemically incompatible so that fertilization does not occur. Postzygotic reproduction isolation acts after fertilization. The embryo may not develop normally, or the offspring may be unhealthy or infertile as adults. In all these ways, reproductive isolation may prevent the gene pools of the two groups from mixing, allowing them to continue on independent evolutionary trajectories. Petropatric speciation closely resembles allotropic speciation. New species arise in isolated smaller peripheral populations that are prevented from exchanging genes with the main population. In parapatric speciation, small population size would make speciation a more likely consequence of the geographical isolation, since genetic drift acts more quickly in small populations. Genetic drift and, perhaps, strong selective pressures would cause a rapid genetic change that could be sufficiently substantial to bring in a new species. Because the extent of genetic drift can't be measured after the fact, testing the hypothesis of this mode of speciation is very difficult. Unlike in the case of allopatric speciation and parapatric speciation, no physical barrier is involved in the other two kinds of speciation. In parapatric speciation, the population members share the same environment but fail to mate randomly. individuals are more likely to mate with their geographic neighbors than with individuals in a more distant part of the environment. In this mode, divergence may happen because of reduced gene flow within the population and varying selection pressures across the population's physical range. Sympatric speciation occurs when a subpopulation that occupies the same niche as the remainder of the species develops a unique mutation that prevents it from mating with the original population. This speciation phenomenon most commonly occurs through polyploidy, in which an offspring or group of offspring will be produced with twice the normal number of chromosomes, where a normal individual has 2 copies of each chromosome. Diploidy. Their offspring may have 4 copies. Tetraploidy. Since a tetraploid individual cannot mate with a diploid individual, reproductive isolation would be created, which might eventually lead to speciation.